My name is Yuchen. I'll introduce the paper submitted to iMeta. The title is The Impact of School Heavy Metal Exposure on Children's Gut Microbiota, the Mediating Role of Environmental Microorganisms. Heavy metals are toxic and harmful pollutants that can affect the school environment and the exposed children's health. This study collected dust samples and children's fecal specimens, and performed gene sequencing. We used extreme gradient boosting to determine the impact of heavy metals on environmental microorganisms and gut microbiota, and the relative length of the quadrant and fourth corner analysis to explore the relationship among the three components. We found heavy metal pollution existed in classroom environment, and lead and copper significantly impacted environmental microorganisms' community structure. Although non-significant associations were observed between heavy metals and gut microbiota, fourth corner analysis demonstrated the associations were significantly mediated by environmental microorganisms. Both heavy metals and microorganisms in the environment can disrupt the microbial community structure in the intestines of exposed children. Manganese exhibited the highest concentration of metal elements, while cadmium had the lowest concentration, and strong positive intercorrelations were observed among all heavy metals. Significant differences in the composition of heavy metals were determined in dust from different sites. For the pollution evaluation, the pollution load index of indoor dust was significantly lower than that of air conditioner filter and outdoor. In addition, the average geoaccumulation index and enrichment factor indicated a heavy pollution of cadmium. As for indoor environmental microorganisms, at both phylum and species levels, clear differences in relative abundance were observed across varying pollution levels. Actinobacillus pleuropneumonia showed higher abundance in heavy pollution. Alpha analysis and PCOA results showed no significant differences across three pollution levels. LEFSA analysis highlighted that cyanobacteriota was a key taxon in heavy pollution. All heavy metals posed no health risks yet. Alpha analysis and PCOA results showed no significance across three pollution levels. The LEFSA analysis indicated that Escherichia shigella showed higher abundant in moderate pollution, Leuconostocaceae and Weissella showed higher abundant in heavy pollution. By examining the metabolic pathways, interactions, and environmental response mechanisms of microorganisms from a comprehensive perspective, we can clarify their critical roles in a wide range of biological processes and identify potential adverse effects on children's health. Lead and cobalt significantly affected environmental and indoor microbial structure. The SHAP results showed that cadmium affected the Shannon index at the phylum level, copper affected the species level across all sites, and cadmium affected the indoor sample's Shannon index. All metals negatively impacted microbial diversity at the phylum level and species level, except for nickel and copper at the species level. Correlation analysis indicated that the correlation between key heavy metals and pollution-associated microorganisms was stronger than that with site-associated microorganisms. For gut microbiota, lead and cadmium presented significant impact at the genus level. The SHAP analysis indicated that chromium and cadmium affected the Shannon index at the phylum level, manganese and chromium affected at the genus level. Only collinzella showed significant positive correlation with manganese and vanadium, only copper significantly affected microbial KEGG function. Overall, the correlations between specific heavy metals and gut microbiota or microbial functions were more subtle than that of environmental microorganisms. In the relative length of the quadrant analysis for samples and species abundance matrix, as the pollution level increased, the dots moved towards the third quadrant, where 47 upregulated environmental microorganisms were located. Nickel, cadmium, chromium, manganese, vanadium, and gut microbiota Weissella also moved towards the third quadrant. Cadmium significantly correlated with Erysipelatoclostridium and Weissella, lead significantly correlated with Bacteroideaceae, Bacteroids, Intestinobacter, and Butyrococcus. Procrustes analysis also confirmed a strong correlation between the gut microbiota and heavy metals. Summary: this study indicates the presence of heavy metal pollution in schools, and its significant impact on the structure of environmental microbial community. Lead and cadmium were found to have pronounced effects on environmental microorganisms. Heavy metals and environmental microorganisms jointly affect the gut microbiota structure of exposed children, with environmental microorganisms playing a crucial intermediary role. Promoting healthy school construction is vital to establish and enhance monitoring systems for heavy metal pollution, raise environmental health awareness, and protect children's well-being.